Dear students, the topic to be discussed today belongs to the course Mmb205, and this is the last topic of your module 2, Japanese encephalitis. Japanese encephalitis, or JE, is a viral borne disease. Particularly, it is an inapparent to acute arboviral infection of donkeys, horses, pigs, including humans. It's a zoonotic viral disease. It is infecting mainly animals and incidentally to human. Caused by a flavivirus known as Japanese encephalitis virus, JEV, transmitted by the vector mostly mosquitoes belonging to the Culex species, affects both the human and the animals, and more than 3 billion people are at risk of developing this particular disease. JE virus actually belongs to the group 4 of Baltimore classification, family Flaviviridae, genus Flavivirus, species Japanese encephalitis virus. It has a genetic material, single-stranded positive sense RNA, so this should be genetic. Now, about the morphology, this is a spherical type of virus, enveloped virus, having diameter 40 to 50 nanometers, very small virus covered with a protein envelope of glycoprotein E. Here you can see it's a dimer, glycoprotein E, and a membrane protein here, M. Closely related, this particular virus is closely related to West Nile virus, West Nile fever virus, and St. Louis encephalitis virus. Here the envelope of this particular virus is actually helping the virus to enter into the host cell, and based on the envelope gene, there are 5 genotypes of this particular virus. They are marked as 1 to 5. Now, about the genome organization, if we look into the genome organization, it is a single-stranded positive sense RNA, and total 11,000 bases. So, particularly, this particular virus contains 3 structural genes, as C encodes nucleocapsid, TRM, chaperone, are the M protein, and EG, the largest structural protein, main target for humoral immune responses. And they also encodes 7 non-structural proteins. They have 7 non-structural genes also: NS1, NS2A, 2B, NS3, NS4A, 4B, and NS5. Where NS1 actually responsible for replication and regulation of innate immune response. NS3, NS4A, and 4B act as serine protease and RNA dependent, the synthesis of serine protease and RNA dependent RNA polymerase enzymes. So this is about the organization of JE viral genome. Now, how this particular virus replicate? So, as I have already mentioned, that this is an enveloped virus, and this envelope, both M and E protein, they are actually responsible for viral entry. Here, the virus entering the host cell via receptor-mediated endocytosis, and then after entering into the after attachment or absorption at the cell surface, they will enter into the cell via endocytosis, and then we can see a membrane fusion. And this fusion will just help the virus to enter within the cell, and then there will be uncoating of the viral capsid as well as envelope. Then they will release the genomic material, means the positive sense single-stranded RNA, and this positive sense single-stranded RNA will then go for RNA replication and subsequent transcription and translation. So after a translation process, they will go for assembly and maturation in association with the endoplasmic reticulum and Golgi apparatus. After maturation, they will come out from the infected animal cell by the process known as budding. So this is the overall replication cycle of Japanese encephalitis virus. Now, what are the hosts? Mostly the hosts are horses and donkeys, and the primary affected domestic animals like horses and donkeys. They are the dead-end host and very susceptible host. Then pigs actually acts as an amplifier of the virus, producing high viremias which infect mosquitoes that are acting as a vector. So the natural maintenance reservoir for JE virus are the birds like herons and egrets. These pictures you can see about the herons and the egrets, the birds that are actually the maintenance reservoir, acting as a maintenance reservoir. So the donkeys and horses are the domestic animals. They are actually acting as a dead-end host. Pigs are the amplifiers, and the birds are like herons and egrets acts as maintenance, natural maintenance reservoir of J virus. So the pigs are actually acting as amplifier. So here, due to viremia, so from their blood, this particular virus will come to the mosquitoes. So mostly a Culex species, Culex Tritini Orynchus, a rice field breeding mosquito, is a major vector for JEV transmission in most of the countries. Other vectors include like Culex annulirostris, that is the Australia-specific strain, then Culex gelidus, Culex fusocephala, these are India, Malaysia, Thailand born species, and then Culex visnoi, this is the India-specific species of Culex mosquito. So other mosquitoes such as Aedes and Anopheles are also considered as vector for JE, but the condition is very rare. So mosquitoes are responsible for pig cycle as well as bird mosquito bird cycle. So mode of transmission is that human gets infected by the bite of the infected Culex mosquitoes. So man-to-man transmission do not occur, and the infection does not spread from man belonging to the means human beings to mosquitoes. So the reverse cannot possible. Mosquito to human is possible, but from human being to mosquito, the infection doesn't spread. And no report of accidental laboratory infections as well as congenital infections or transmission from infected organ donor is available. This is a life cycle of Japanese encephalitis virus. Here you can see bird, mosquito, bird, or else bird and mosquito, pig, mosquito, pig cycle, then to human. This is the life cycle of JEV or Japanese encephalitis virus, and this is a natural cycle. So birds are the herons or egrets are the natural reservoirs, And so bird mosquito bird cycle, or else bird pig pig mosquito pig cycles, they are the amplifiers, and from there humans are getting infected. So this is the natural life cycle of JV. Next one is the pathogenesis. So virus enter the body through the bite of the insect vector, means mosquitoes, Culex mosquitoes specifically. So after multiplication in the reticuloendothelial system, viremia of varying duration ensues, and virus is transported to target organ, um, uh, like brain, via blood, and virus proliferate and damage the neuronal tissue that elicit nervous manifestation, that elicit nervous manifestations. So pathogenesis means Japanese encephalitis virus infection causes a neuronal damage in the brain through crossing the blood-brain barrier by passive transport across the endothelium, and monocytes and macrophage carriers of the virus in the central nervous system deterioration of blood-brain barrier stability. So JV infection activates microglia, while causes a raise in the level of pro-inflammatory cytokines such as interleukin 6, tumor necrotic factor alpha, MCP1, etc., involved in inducing the neuronal death. Next is the clinical features. So here the incubation period ranges from or between 5 to 15 days. Most of the infection occurs in childhood. Adult infections are very less frequent, and mostly the disease is asymptomatic or maybe mildly symptomatic. So about 1 in 250 infections show symptoms of encephalitis, and 30% of persons who show symptoms die from the disease. 40 to 50% of persons who survive suffer from permanent neurological defects such as paralysis, recurrent seizure, or inability to speak. So this is a very dangerous type of virus. So clinical features. So in case of the symptomatic patients, the disease manifests in 3 different phases. One is the acute prodromal phase. So before a central nervous system entry by the virus, so there may be fever, GI infection, gastrointestinal infections or disturbances, headache, etc. May be noticed. And in case of acute encephalitic phase, so after CNS entry by the virus, so rapid onset of high fever, neck stiffness, then seizure, spastic paralysis, and eventually death may happen. And in case of recovery phase, so complete or partial recovery with neurological deficit or defects and cranial nerve palsies can occur. So these are the clinical features after the infection of Japanese encephalitis virus. So to go for diagnosis, so sample collection and handling and choice of specimen is very important. So for the detection of antibodies, mostly IgM antibodies for Japanese encephalitis virus, so detailed information should be there in store. So you have to collect actually cerebrospinal fluid, minimum 0.5 ml CSF is required. And this collected CSF should be stored in minus 20 degrees centigrade, so they're minus 20 degrees centigrade. And blood specimen collected collection should be 5 ml for all the children and adults, and 1 ml for the infants and the younger children. So diagnosis, mostly ELISA test, enzyme-linked immunosorbent assay for IgM specific antibody for JV should be assessed, and then the PRNT test means plaque reduction neutralization test that differentiate the J virus from the other sorts of viruses. And if you are sure, you can go for further tests, that is reverse transcription polymerase chain reaction to detect the RNA expression. And it should be noted that IgM ELISA is the method of choice, provided samples are collected 3 to 5 days after the infection. So treatment, there is no such specific antiviral medicine available for JE. So clinical management is supportive. Fluids and electrolyte balance is a must during the acute phase of the disease. So seizure management as well as airway management are very crucial for the treatment. Next, control measure. This is very important. So it involves two strategies. One is the control of the reservoir. Second one is the control of the vector. So control of the reservoir, if they are birds and other vertebrates, animals can act as natural reservoir, but it is practically impossible to take care of these reservoirs because birds are migratory. So to take care of this, it is practically impossible. But pigs that are actually acting as amplifier, so we can take precautions. So pigs also act as an amplifying host. So pig rearing should be discouraged in areas where rice cultivation is widespread. So this should be followed. And the second one is the control of the vector. So insecticide spraying is out of option as vector mosquitoes breed in paddy field. So eco management of paddy field can be done, means alternate wetting and drying instead of irrigation system. Ultra low volume insecticides can be sprayed by fogging, has been found helpful in some extent, to some extent. So sterile melt technique is a novel approach. So so we can go with this particular technique, sterile melt technique, rather than using insecticide spraying. So prevention, 4 types of vaccines are available for the use of JE vaccine. One is mouse brain derived killed vaccine, then cell culture based killed vaccine, live attenuated vaccine, and live chimeric vaccines. These are the 4 available vaccines available in market for JE. So mouse brain derived killed vaccine, Nakayama and Beijing strains are used here. So widely used vaccine in the past, primarily dose followed by 2 boosters, but this is expensive and mostly ideal for the travelers. But this particular vaccine has severe adverse effect. That is why this particular vaccine was banned from 2007 in India and in many other countries. So live attenuated vaccine, also called SA-14-14-2 vaccine. So presently used in India, 2 doses of 0.5 ml subcutaneously, and safer up to 15 years of age. Not recommended for the adults. So only for the children up to 15 years of age. Then GenVac. So varicella derived purified inactivated vaccine, indigenous vaccine, made using strains obtained from Kolar, district of Karnataka. So 2 doses intramuscularly, 20 days apart, 28 days, in fact, 28 days apart for routine immunization, and single dose of 0.5 ml during epidemics. So 98% seroconversion after 2 doses intramuscularly. This particular vaccine was launched officially in October 2013, and it is available in market, but not yet included in routine immunization schedule by the government of India. That's all. Thank you.